so this was the question of what is the new species of the week. This was the clue that I provided to you. And the prediction results are in 10 votes on a copepod, 10 votes on a crab, 0 votes on a squid, 0 votes on a fish, 0 votes on a bee, 0 votes on an ant, 0 votes on an octopus, 0 votes on a squid, 25,000 votes on a spider. The correct answer is, folks, a crab. Is what I would say if crab was the answer. However, it is a crab spider. This was the beauty. No, Entropic, don't worry. It is a spooter. It is a spooter. It is called a crab spider. And spider warning for those who don't like spooters. It is a very, very, very bizarre looking arachnid, I think. It is a very, very unique shape, right? It's very exotic looking. This is the dorsal part of its body. And there's like all these little nooks and divots and crannies on it. They also, this paper actually did both male and female. Look at that. Why does this matter? Because in a lot of papers, when we talk about the new species of the week, they don't do both male and female, right? It's only, like, a lot of times it's male, and they talk a lot about the different phalli or the phalluses. And the reason for that, honestly, is because they tend to have a lot of different shapes. And those tend to be like an identifying factor. Or there are some animals that are easier to find, like the males, than it is to find the females. And that is in part because it's like the females, you know, will be more likely to hide because they're also providing brood care. And so you're not seeing this. What werewolf, they're obsessed, yes, but it's also they're structurally very different, if that makes sense. Yeah, so Morris, this specimen is damaged. They even mentioned it that not in the figure but in the paper that they mentioned, Morris, that it looks like that they had some breaking in terms of preservation of this. They ended up putting it into alcohol for the final means of preservation. Well, let's take a look at it, Entropic. We'll just go through the paper. This is another Zuki's paper. Zuki's is a journal, honestly, y'all, that is known for publishing new species. If not a brand new species, a reclassification. So if you're looking at the paper and you end up seeing like, oh, this isn't a new species, but we're redefining it as a new species. Or sometimes they also merge ones together. Like, we used to think that this were 3 different species, and now we think it's actually one species. They'll have papers like that as well. So that's the kind of journal that this is the kind of paper that gets in there. And the kind of stuff that they publish. But this is a brand new genus, not even new species, brand new genus, and they describe 2 species within this new genus, right? So you get a BOGO today, y'all. You get a 2 for the price of 1. And it is from China. It's just really cool. There's like, it's so different. It's a brand new genus, right? And there's 2 species that they're able to describe here. And so remember, if we break down the classification system of what is a species, it goes kingdom, phylum, class, order, family, genus, species. And they mention that there are 3 different families or 3 spiders of a certain family from China. And now they find within that family, there is a new genus. So in the breakdown of what is like the taxonomic, map, if you will, they now make under the same family where there already had been identified spiders. They say, oh, look, new genus and 2 new species involved. So again, and if we just break down the abstract, so remember, y'all, the abstract is the summary of the paper, what you're about to get, the meat and potatoes, if you will. So going into the fact that they have found, based on the polyp, so some of the structural processes of these spiders, that they're uniquely, visibly different. So there are those striped patterns that actually some of y'all in chat were like, it looks like there's different kinds of freckles and there's this striping pattern that looks interesting. That's what they saw too. And like, oh, that's really, that looks different. Maybe that's a new thing. And then what I really like, Yoga Garrel, they did scanning electron microscopy of the spiders. Y'all, I am a sucker for scanning electron microscopy. It is really, really high resolution imaging where you're zooming in and checking out like all the detail that you possibly can. It is notoriously difficult to do on a spooter. Why? Because a spooter dries out. On an insect with an exoskeleton, like an ant, that hard exoskeleton, it might dry out on the inside, but that exoskeleton ain't changing, right? If you can dry it out, you can do whatever you want with it, and that exoskeleton will stay the same. A spider is much softer. Yeah, there's an exoskeleton covering, but it's not that hard casing as what you might see on an ant. So in order to get a really really good image, you have to undergo a special preservation process. This is true for other scanning electron microscope things, imageries. You can do scanning electron microscopy on a cell. A cell, right, if you put it out on a dish without any liquid, will dry out. So it's not the beam destroys a specimen. Well, it would in Tropic. You have to dry out the sample and coat it with either platinum or gold. And then what the microscope does is that it launches a beam at the specimen, and from the specimen you get a bounce. The beam bounces and is detected by a sensor, and that's how you get your images in the scanning electron microscope. If you don't have the covering of the metal, you get what is called supercharging. Supercharging comes across as a very white image. No detailing whatsoever. It's just a white, descriptionless splotch. And yes, Morris, you are correct. So it's the drying part, because Morris, if you're trying to get information about this new animal or any animal that you want to measure, if it's dried out, can you really get that measurement correctly? So it's not just the coating. The coating is in part attenuates this, but you still have to be able to dry it out, the specimen, in a way you can dry it out, rehydrate it, and dry it out and rehydrate it, but then where the structure is maintained. So even though the innards of it can now be dry, you're maintaining the structure of it. And that's the critical part. So exactly. So Morris, the same thing happens if you take a cell and you put it on a plate. If you dry out that cell goes from being a nice circle to, right, it looks then like a banana instead of a circle. You can attenuate that as well with a process of drying out, rehydrating, dry it out, rehydrating, back and forth, and that's what keeps the information intact. That is not something you have to do with an animal that you're looking at with a very strong exoskeleton, because it can't dry out because it's, like a chitinous outside, and that's just keeping everything intact. And then here we go. So a common name, this is what I was going for in Tropic with the naming system, is that it is a running crab spider. It is a crab spider that we've got here. Can you use alcohol to dry it out like an IHC? So Matt, so what Matt is saying, y'all, Matt is a scientist. IHC is immunohistochemistry. That's some of the glowing images that we've looked at in the past. For this, Matt, you're drying out specimens for scanning electron microscopy. You don't use alcohol. You have a very set list of ingredients that you utilize. And it's also like the microscope dependent too, of like what you're going to end up coating with. And so when I was doing it, I do not remember the exact chemical, but it was not regular alcohol. It was something that was super corrosive, and I had to apply it under a fume hood and wearing gloves, obviously. Like, I mean, you should wear gloves all the time, but for this, it was like, yeah, you really need to wear like that kind of flavor. And that was for a certain cell type that I was trying to get, to be able to dry out the cell, and then rehydrate it and keep that contents preserved. Didn't need to do it for the ant exoskeleton, but did need to do it for other stuff. And yeah, Morris is saying right, to avoid raisins. So let's just go ahead and start looking at the images. I think they're really pretty. This is one of the two new spiders. Remember, y'all, they discovered a new genus of spider. A genus is that before classification of a species. So there's a genus and then two species on there. Yep. So Morris, this is one of the two new ones. Look at this. This is what I find so cool. Look at the abdomen. It's got some really nice patterning on it. And this is what the scientists were like. This is like common, let me go outside and look at stuff science, right? I'm outside, I see a funny looking weird thing. Let me go figure out what it is. And apparently they don't have anything that looks like this. That was the cue off to figure out that this is new. The other thing that they mentioned is that the palps, or some of the organs that are utilized, like in this, that's unique to the spider, are also looking different. So here in C and D, this is actually the copulatory duct. So these are the mating ducts used when the animals are mating, and so they're even able to see this visibly without like a high-powered microscope, like a nice camera. This is a spider part that we looked at earlier today. Oh yeah, here are the palps, like a nice zoom in. I want to show you all this picture too, because I thought it was super. Look at that. Look at the textures on the palps. Like, they're just really pretty. Nice little, like they didn't hypothesize like if there was any almost point to these structures in terms of looking like this, presumably because they did measure and there are sexual dimorphisms, so there are fundamental differences between male and female. It might be some flavor of sexual selection, and it's just attractive to certain kinds of spiders. Oh no, these are photos, Morris. These are straight up high-res, high-powered photos. Ramon, this is one of the new species of the week. This is a new species of spider, right? Mark, it's super pretty. Like so what this imaging is, to make it this pretty, Morris, it's called image stacking. You hook up the image or the microscope to a camera and you image focal planes. So a little something is in focus. You then turn the focus onto something else, something else, something else. And you keep doing that over and over and over and over again until you have a bunch of images. And then you put them all together and it makes it all in focus, which I think is a really powerful technique. That's why this looks so crisp. You have to take all the images all set, like nothing can move. It all has to be at the same level. Yeah, I just thought it was a really cool set of images they're able to. Let's get to the business end of things. Here's that new species that we started off. This was your clue, if you will. That was what we clued in on, the yellow green colors, and this is their palps as well. Has a similar like wavy structure in it, but again, different color, looking quite unique. The little freckled spots, as y'all were saying, they've got on there. I'm actually not sure about the freckly spots. Sometimes that could be an effect during development, where what actually ends up happening is that it's almost a immune response where the cells were melanized and release this melanin pigment, and then it's just something that's maintained in adulthood. It could be something like that, or it might actually be on the exoskeleton. It's not entirely clear to me of where this is located based on the way they're reporting the data, but it's just something to kind of keep in mind, and I think it's pretty heckin' cool. Let's go to the here it is. All right, so here they look at the palps, and Morris, this is the beauty right here. So because they're arguing that the palps are unique structure and they're looking very unique in this particular species, they went to that scanning electron microscope set of images, and these are SEM images. So here, these are all entropic coated in metal now, and it is not easy to do this because so it's sputter coating entropic. So what that means, it's a light dusting to coat it in metal. And that light dusting is difficult because if you overcover, you lose a lot of these details. Like these little hairs that you see here, if you overcover it, you lose that level of detail. But if you undercoat it, you get something like this. Do you see entropic how here it's it's much whiter here than it is on the rest of the specimen? It's not really distracting here, but it is much whiter, right? That is called supercharging, where it's undercoated, and you can imagine where if the whole specimen is that white in color, you're not going to get any detailing. So it's a lot of times it's that process of coat it a little bit and then put it in their microscope and see what it looks like. The difficulty with that is the coating process is in a vacuum sealed chamber. The microscopy itself is in a vacuum sealed chamber. Vacuum sealing on its own, if depending on how your specimens are mounted, could cause some damage too. So you don't necessarily want to be doing that process over and over and over again, because that could affect what you end up getting. Does that make sense? But yeah, so this image in Tropic is just really beautiful. Like this series where you can even see the individual like fibrous bristles of the hairs on lighter sample, and it's just a great way to kind of highlight what they're trying to image, and like what they're measuring. I will say in Tropic, this image here, on J, if y'all see these little lines, so these little lines are actually a result of having to take different images, because what this suggests to me, so normally in Tropic you will see the scan lines when you're imaging. When you're looking at a preview of one of these specimens, it just goes, and you see that over and over again, and you see those lines, and it doesn't look great. But when you take the image, what you end up doing is you look at it in low res because it's faster, and then you take a high res image, which takes anywhere from 3 minutes to an hour, depending on how high res you want it. Here, though, they had to change the laser intensity between the different stacks, because my guess is that it was supercharged and they weren't able to get stuff in correct focus, and that's why there is these scan lines, is because they had to change around that laser intensity between different layers. Morris, these are the scanning electron images. They're like really these are the ones where it's like the animals dried out coated in metal and getting a really, really nice, beautiful close look. Yeah, really, really pretty. Look at this, a beautiful set of images, Morris. Just again, y'all, these are zoom-ins of the new spider species that were identified. And here, like, these are how they're taking some of those measurements. There are some scan lines on here. This is another example for entropic of that supercharging, where the different layers have different amounts of coating, and that's why you're getting this these charging lines, right? Still really, really beautiful images. Like the Miss Capri Kel, what's really cool is it's a new genus. So it's not just they discovered 2 new species of a brand new genus. Because this was in southern China. They had known that there is about 2 to 3 families of spiders in this area, and they were just looking. They're just using their eyes to look for something new. And all of a sudden they find a brand new looking spider. So visibly it looks different. And when they did the sequencing, they found it belonged to a brand new genus. Like they were different enough to be not an existing genus, but a brand new one. In terms of venomics, we'll keep scrolling here, but like just to show you, a lot of what they did was measurements, a lot of really nice imaging. They even looked through and talked male and female. They've got measurements, right? 2.5 to 4.5 millimeters. The male has more black spots than the female. They measure the different eyes, the different numbers of it. The male polyps, that's the male reproductive organ, because it looked so fundamentally different from other ones, but they do look at both male and female. And then here are, again, those like really, really high res, beautiful images that they've got. The scanning electron microscope, again, of spiders. Now, the reason that this is cool, again, as we were mentioning, spiders dry out. Actually, also really cool, you can see here, this in the background is a EM stud. So in order to preserve this, you actually take a piece of double-sided carbon tape. It is an extremely sticky tape. Put it onto a stud, put the specimen on the stud, and then you do the metal coating. And you can kind of see that texture there is that background of the stud and the tape. There is, and you can almost see like some stuff is stuck to this base. So that's like that stickiness of the tape in the back here. So they could have taken it higher res, Morris, which would have allowed us to zoom even more. Higher res images take longer because the microscope spends longer on each pixel when taking the image, when the sensor is pulling in the images. So these are the distributions where the new species of spider was identified. Nice distribution. They did a little bit of sequencing collection in this, and the sequencing studies here in this paper were very minimal. So instead of being the sequencing that we've talked about in the past, where it's like a whole genome sequencing, this is that very, very small region of the mitochondrial genome that is commonly said to be like the different zone, right? That's indicative of evolution and a lot of different changes. And that is what they sequenced. Not a full genome, right? The argument though here is that it is different enough in these specimens to be like, they look so different from anything that has been described before that it's already probably a new species, right? So the way that they did the imaging and the measurements, probably a new species, just based on that alone. And then they did the sequencing on that subregion of the mitochondrial genome, and they're like, all right, this is then surely, and don't call me Shirley, a new species. That's kind of the argument that is being made here. Hopefully that makes sense. So that's the paper, y'all. Let me just show y'all. This is not the new species, but this is a member of that crab spider group. That's just kind of what they look like in the wild. Like, of this family, I actually know how to... I have a lot of pronunciation difficulty with the Latin on this one. They are not part of the same family. Let's see, but we can see proximity of Philodromidae, and there's an image. Doesn't have it. Why would this come up? This genetic tree is terrible. Terrible trees here. All right, there's Arachnidae. Yeah, I can't find a tree with both of them. So it looks like Turgis, the split point is here. We don't actually know how many millions of years ago this was. So usually, y'all, if we are analyzing the tree of life, this is the kind of split point that we're looking for using this. And the split point, right, it'll tell you most common species to these are at this point. This one goes off in this direction. This one goes off in this direction. And we see some element of time. We don't see it, but let's see the running crab spiders. Actually, it looks like Turgis within that branch. It looks like they're super, super deep on this. This isn't going to the species level Turgis. This went to the family. So it looks like it's quite a jump in terms of evolutionary time. And this again this is just the family. So Turgis, it wasn't us going from a particular species, it was going from the family of the orb weavers to the family of the crabs, the crab spider, running spider. See, there's a lot of these species, and you can move in further, but they do look pretty separate. And it's cool. Actually, we had a member from chat who came in who said that jumping spiders were cool. It looks like that these crab spiders, their next door relatives are jumping spiders, which is kind of cool. So whoever said that in the chat, I forget now who had mentioned it, but I was like, wow, that's like a really, really cool like observation. And it's like by evolutionary relationships too. Like that's a heck of an observation right there. Cool.